Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today, we are talking about steroid hormones, steroidogenesis, its applications, and biosynthesis. This is our second lectures on steroidal hormones, in which we will discuss the synthesis of corticoids, cortisol, and aldosterone. We will also discuss how different kinds of hormones are derived from the cholesterol, which is the basic precursors, as well as we will discuss the functions of different kinds of steroidal hormones. Let's see the synthesis of corticoids. Corticoids are divided into gluco and mineralocorticoids hormones. The predominant human glucocorticoids, cortisol, is synthesized in the zona fasciculata of the adrenal cortex. The synthesis of cortisol involves hydroxylations of progesterone at the 17 alpha carbon 21, that is represented by CYP21 and 11B. CYP11B1 are the positions which are mentions upon carbon 21 progesterones. The synthesis of cortisol is under the control of an anterior pituitary hormones, ACTH, and negative feedback mechanism in which elevated cortisol suppresses the release of ACTH. The dominant human mineralocorticoid is aldosterone, which is produced in the zona glomerulosa of the adrenal. The synthesis of aldosterone involves the synthesis of corticosterone and subsequent hydroxylations and oxidations at carbon 18 to yield aldosterones. The synthesis of aldosterone is regulated directly by levels of potassium and indirectly by the effect of sodium levels and blood volume on levels of angiotensin II. Synthesis of cortisol. Cortisol is the example of corticoids. Let's see how cortisol is synthesized. In the first step, pregnenolone is converted into 17-hydroxy pregnenolone in the presence of CYP17 alpha hydroxylase enzyme. You can see in the first reaction the pregnenolone, in which the acetyl group is present at carbon 17 position, is converted into 17 hydroxy pregnenolone in the presence of enzyme CYP17. In the second molecule, there is a presence of acetyl group at carbon 17 position in addition with hydroxyl group at carbon 17 position too. So, in the next step, the 17 hydroxy pregnenolone is converted into 17 hydroxy progesterone in the presence of 13 BHDS enzyme, which is then converted into deoxycortisol in the presence of 21 hydroxylase enzyme. You can see in the second step of reaction, 3-BHSD enzyme affects the 17 hydroxy pregnenolone, which is converted into 17 hydroxy progesterone. In the progesterone, you can see over here in this structure, there is a presence of acetyl group and hydroxyl group at carbon 13 positions, while acetyl group at carbon 17 position. So, this molecule is further converted into 11-deoxycortisol. In deoxycortisol, at carbon 3 position, there is the presence of carbonyl group, and at carbon 17 position, there is the presence of methoxy group addition with the carbonyl group. Further, 11-deoxycortisol is converted into cortisol in the presence of 17 beta hydroxylase enzyme. In the reactions over here, you can see 11-deoxycortisol is converted into cortisol in the presence of CYP11B1 enzyme. The difference between both the structure is that There is a presence of hydroxyl group in the cortisol at carbon 11 position and further hydroxyl group at carbon 17 positions, in addition with methoxy group which is attached to the carbonyl group at carbon 17 position. There is also a presence of double bond or unsaturation at carbon 4 position in the cortisol. So, in both the structures, the difference between the function moieties is at carbon 11 position, carbon 4 positions, and carbon 17 position. So, this is the method for the preparations of cortisol. Synthesis of aldosterone. Aldosterone is synthesized in the body from corticosterone, a steroid derived from cholesterol. The production of aldosterone in adult humans about 20 to 200 micrograms per day in the zona glomerulosa of the adrenal cortex is regulatory by the renin-angiotensin system. So, in the first step, the cholesterol is converted in pregnenolone in the presence of a specific enzyme. In next step, the pregnenolone is converted into progesterone in the presence of 3-DHSD enzyme, which is generally represented as 3-beta hydroxysteroid dehydrogenase. After that, progesterone is further converted into deoxycorticosterone in the presence of CYP21A2 enzyme, that is written as CYP21A2 hydroxylase. At the end, deoxycorticosterone is converted into corticosterone in the presence of CYP11B1 beta hydroxylase enzymes, and then it is converted into aldosterone, which is our target compound, in the presence of CYP11B2 aldosterone synthase enzyme. Let's see the synthesis of different hormones from cholesterol. First of all, we will discuss the structure of cholesterol. The total number of carbon atoms of cholesterol consists of carbon 27 atoms, in which the hydroxyl group is present at carbon 3 and double bond is present at carbon 5. The hydrocarbon side chain, which consists of total number of 7 carbon atoms, is attached at carbon 17, in which two methyl group are present at carbon 25 and carbon 20 positions. In addition, two extra methyls are present at carbon 18 and carbon 19. So, this is the common structure of cholesterol, or which is generally known as cholest 5-ene 3-beta ol. Now, we will see from where the cholesterol is came out and how it is used for the synthesis of different types of steroidal hormones. In first step, the cholesterol is uptake from LDL-C via LDL-R, that is called low density lipoproteins receptor, is a mosaic proteins of 839 amino acids. After the removal of 21 amino acid signal peptides that mediates the endocytosis of cholesterol rich LDL. It is a cell surface receptor that recognizes the apoproteins B100, which is embedded in the outer phospholipid layer of LDL particles. In humans, the LDL receptor protein is encoded by the LDLR gene on chromosome 19. It belongs to the low density lipoprotein receptor gene family. It is more significantly expressed in bronchial epithelial cells and adrenal glands and cortex tissues. The second way from where the cholesterol is isolated, that is de novo synthesis. De novo synthesis refers to the synthesis of complex molecules from simple molecules such as sugar or amino acids, as opposed to recycling after partial degradations. So during deodorsynthesis, cholesterol is synthesized, which is used for the preparation and synthesis of different steroidal hormones. For example, nucleotides are not needed in the diet as they can be constructed from small precursor molecules such as formate and aspartate. The third way from which the cholesterol can be used for the synthesis of different steroidal hormones is cholesterol, which is cholesterol esters, or cholesterol molecules with long chain fatty acids linked to the hydroxyl group. They are much less polar than free cholesterol and appear to be the preferred form for transport in the plasma and for storage. So, the cholesterol in the first step is converted into pregnenolone by using 450-SEC enzyme. This is the first step for the synthesis of steroidal hormones, that is pregnenolone. The difference between both the structure is that there is a presence of hydroxyl group at carbon 3 positions in beta form as well as acetyl group at 17 positions in the pregnenolone structures.